this work?
Yes, let me show it. Basically, and I think like that. No, it's the other way around.
Okay, where?
No, behind your ears.
Oh, okay. Oh, it's
Okay, can you hear us online? Yes? Ah, great. And also spotlight the presenter. Yeah, thanks. So hello, everyone. Thanks for joining. We're very happy to have here with us today Dr. Serghei Mangul. Dr. Serghei Mangul holds positions as a Director of Challenges in Benchmarking and assay-based Bionetworks and an Assistant Professor of Clinical Pharmacy and Computational Biology at the University of Southern California. He specializes in the design, development, and application of novel data-driven computational approaches to accelerate the diffusion of genomics and biomedical data into translational research and education. I guess he will also tell more for us about his work. Thank you. The floor is yours.
Yep, thanks so much. So really excited to be here. And then if you want to talk to me later about Sage Bionetworks, so now I'm directing the challenge and benchmarking platforms, how to organize the challenges, how to run benchmarking. Happy to talk about that. So today I would like to talk about adaptive human receptor repertoire and see what, as computer scientists, we can do there, right? Like where are the opportunities? And just giving you a heads up, I think we have a lot of open problems in immunology, in particular adaptive immune repertoire. And I think we have a limited workforce as it comes to computational research, right? So there is a need, and probably pressing need, to develop better methods, to create better databases. So that's, I think, good news for us, so we can contribute to the field. So just to get started, I would like to give my definition of bioinformatics and then like the vision we have as we develop the method. So you can imagine, or we are imagining as bioinformatics is that frontier between the noisy data sets on the left, which are large, but not only large, but noisy. And on the right, it's like meaningful interpretation you would like to have in the clinical settings. And this is what bioinformatics is emerging as the frontier between those two. I think it's bioinformatics, if not now, but it's becoming the center of the current biology, where we're moving to the data-centric world, where data plays a substantial role right across the domains, and in particular in bioinformatics, computational biology. And I think it's absolutely crucial to accelerate the science, to make discoveries. And many, many times the discoveries you hear, that people discovered a biomarker in a certain disease, or they found a therapeutic target, which can be used later. But behind that, right, somewhere was bioinformatics method being applied on the data rigorously with high accuracies resulted in that. I think we also see the change in the cultural aspect in bioinformatics, where the people doing bioinformatics are becoming more and more independent. Because if you think about the historical perspective, right, I think it's quite interesting. 10, 15 years ago, I think bioinformatics was serving the biology to some degree. And we as bioinformaticians were having limited ability to kind of perform the research. We were developing the methods, but let's say it was limited opportunities to apply them and conduct independent research. But things have changed, I would say, with the secondary analysis of the data, right, where we do the data reuse. In other words, usually, right, if you think about biomedical research, you would first generate the data and apply some methods, right? So that's called primary analysis. And this is how it's traditionally done. But that have been challenged by the secondary analysis, where the paradigm is existing data, but novel discoveries, right? And that is more and more happening. And there are many, many reasons why you would reuse the data. The most obvious one is meta-analysis, where you would combine multiple studies, right? Like independently, each research group who generated the data did not have, let's say, sample size sufficient to discover, let's say, genetic association with certain disease. But as you would combine all of those studies the meta-analysis way, that gives you the power, right? And you as computational researcher can make substantial discovery. It comes with the challenges, the computational research, I think, data management, right? The data is large. We need to know how to manage the data. The methods needs to be scalable. fast and not require too much resources to decrease the cost of the analysis. So there are ethical concern, data access, can we access all the data, and how to manage the data overall. So this is the secondary and primary analysis in a simple way. So today we'll be mostly talking obviously about, as bioinformatician, about secondary analysis, but just to give you the perspective, right? Like the primary analysis, this is what traditionally is done, right? Like where you would recruit the biological sample, you will be some cost being involved, and you would do some sort of high throughput technology like sequencing, and you would do the analysis and you will publish the results. So as you publish the results, luckily, I guess for us, that all the data which being associated with the publication is required to be shared, right, in some way. So what happens that we have quite a bit of public data around, which we can use in sitting in those public repositories like SRA and GEO, so we can use, right? So the secondary analysis will skip the data generation part and will directly focus on the computational analysis. Okay. So today, like I was thinking to talk about the particular adaptive immune repertoire, we'll call it AIR, and that's emerging field where we can perform the secondary analysis, but we need to develop quite a bit of accurate methods. We need to develop databases, and we also need to decide what technology to use, right? Is it the conventional technology, which is called in this case TCR, BCR sequencing, or should we think about something else? So the problem with the proper technology, which is called AIRR-Seq technology, we have few samples. And also the ancestry is unknown, right? Like if you think about regular technology, like whole genome sequencing, you can infer the ancestry from that, right? You can, and then you can do cross-ancestry studies, but we cannot do it here. As well as there is few samples, right? So this is the concern for secondary analysis. So the reason you want to do secondary analysis, you would like to acquire a lot of samples, but we don't have it. So perhaps it's not that great to study immunome, right? So the immunome we define as the complete set of recombined receptors, and I will get into that in a second, what we mean by that. And this way, we can think about the conventional technologies like RNA-Seq, whole genome sequencing, which can be superior, especially for population level adaptive immune repertoire profiling, right? So the keyword is population. Since we don't have much data in adaptive immune repertoire field, we don't do population level studies at all, right? So that's a concern, and we hope the secondary analysis and using different technology can change that. So just to make sure we're all on the same page, some background. So if we talk about adaptive immune repertoire, this is the loci located across your genome. So there is Ig, immunoglobulin loci, there is TCR, T-cell receptor loci, there is few of them spread across the genome. So if you zoom in one of those loci, what you can see a collection of the V genes, collection of the G genes, and the J genes. So V stands for variable, D stands for diversity, and J stands for joining. So those genes can be recombined in the random fashion. So for example, we decided to recombine V1, D3, and J2, and there is also the constant region. But you perhaps know that the diversity of the T and B cell repertoire is quite large, it's astonishing, it's 10 into the power 15. So if you look at the numbers, perhaps you would see that that kind of mechanism is not enough to generate 10 into the power 15 distinct sequences, right? Because just we have a limited number of those genes to start with. So another thing, what is happening, we will introduce non-template insertions, like random characters, between V1 and D3, between that junction, and D and J junction. And having that machinery will allow us to generate literally any sequence. And this is the reason behind why the diversity of the T and B cell receptor repertoire is 10 into the power 15. So a few definitions, so we'll be talking about CDR1, CDR2, and CDR3 regions. So CDR stands for complementary determining region. So this is the region, CDR3 in this case will be the most diverse one, right? So expanding that diverse region, and we'll be using that as the definition of the clonotype. So what we're gonna say, that we'll say that distinct sequences of amino acids of CDR3 will be used as the proxy for the clonotype, and the collection of the clonotype of the individual will be called the repertoire. So, and to give that into perspective, so previously we were working with RNA-Seq technology, trying to explain every read which is derived by that technology. And what we noticed that among the sources of the reads which are kind of unmapped, right? So which were not used for initial analysis, let's say variant calling or gene expression profiling. Many things are there, but among them we notice, oh, we have VDJ recombinations, right? Like those things we just saw before. And we said, oh, can we use those reads, which are very unconventional and kind of ignored all the time, to reconstruct the immune repertoire, to reconstruct those CDR3 sequences? So this is the numbers, right? So if you think about typical RNA-Seq analysis, you would have majority of the reads being mapped. In other words, they will be used for gene expression computation. However, there are many, many other sources of reads, and the one we were interested in is recombined B and T cells, right? There is 0.02%, a small number, but we would like to ask the question, is that small number enough to get an idea about the immune response or repertoire of an individual? So in terms of, just to kind of spend some time about RNA-Seq, so if you think about RNA-Seq as the technology, you would have RNA molecules, and you would shred them into the fragment, and if each fragment will be sequenced, and this is, you will get the reads, and presumably having the genome being available, you will map the reads onto the genome across the gene, and you would have gene annotation. So what you can do, you can calculate gene expression. This is how it's done. You just count the number of reads per gene, roughly speaking, and that's your gene expression. So having that gene expression profile available, you can look at the differential expression genes, you can look at RNA splicing structure or assemble novel transcripts. However, what is happening, you would ignore a fraction of the reads and they literally will be wasted. So we'd like to ask the question, can we look at reads which are typically ignored with the analysis and look for the VDJ recombinations and the clonotype in that data? And the intuition behind is that if you take a sample and then you prepare the DNA of that sample to be sequenced, you would and if you have B and T cells in that sample, those cells have their own receptors. So you can think about that as having the separate genomes. And those genomes, those receptors are not part of your reference genome. So that's kind of the intuition behind why we have those unmapped reads. So to be more precise, so if you think about coming back to that particular clonotype, so what you can see that if the reads are mapped in black, right? So if the reads are mapped, they will match the V gene. Why? Because V genes are part of your genome, or at least certain alleles of the V genes are part of your genome. However, the recombination are not, right? So the recombination of V gene and D gene and J gene are not part of your genome. So as a result, those gray reads, which are spanning that recombination, will be unmapped. They will be not captured by initial mapping alignment protocol or method. And as well as the certain V alleles are not present in the genome, as a result, reads will be not mapped to those alleles. So to detect the CDR3 sequence, we come up with a quite simple method, which allows us to avoid the mapping, just to take the reads and search for the CDR3 sequences inside. And this is one example where sometimes it's nice and the kind of it's nice to use prior information as much as possible in this case, and still being kind of non-biased. So what I mean by that. So we know the CDR3, if you go open Wikipedia or in any textbooks, you will see the definition of the CDR3 sequence. And then what it will tell you that it's a sequence always starting with cysteine and always ending with phenylalanine. So you can see two letters, C and F. So that's nice, right? That's a nice thing to know. But the question, how we can use that? So what we did, we took a read and we converted the read in all frames in amino acids. So we did that. And now we say, can we look for that sequence inside, right? And if we do so, perhaps it's kind of would make sense to use the definition, right? So what we're gonna do, we're gonna go from the left to the right until we find the cysteine, if we can. And the same way we do from the right to the left until we find phenylalanine. So that looks like CDR3 sequence, but perhaps we need to think a little bit more about that. So that we'll be calling candidate CDR3 sequence. But we also know that we have V genes that should overlap those flanking regions. So we can do that. We can take every of 550 or so genes, take it one by one, and verify that, right? So if we take V1 gene and match it, we'll not see a match. The same for V2, V3, and that's V3 will be a match. So the same procedure we'll do for the J genes. We'll take every J gene one by one, and we're gonna match the J genes to the flanking regions on the right. So if we have two matches, if both flanking regions will match, then we're gonna say, oh, that's no longer candidate CDR frequency sequence. Instead, that's a real CDR frequency we're gonna report. So sometimes reads are short, right? So if technology gives you 75 base pairs or something, or 50 base pairs, reads are short, so they will perhaps not overlap both V and J genes, but only one of them. So let's say you have a read, and it overlaps. You found a C stain, but no matter how hard you try, you don't find the phenylalanine. It can be a random sequence, right? So no relevant to us. That's one possibility. Another possibility, right? That just was short enough. It was just too short to touch, and to touch the J gene. So as a result, to have the phenylalanine. So let's save it, right? So let's first of all find the match. If we have a match of the flanking region with the certain V gene, we will record it, right? So let's record, and we'll call it partial CDR3 sequence. So we're gonna record all the reads overlapping only the V gene, right? That will be on the left. And on the right, let's record all the reads overlapping the J genes only, right? And having those partial CDR3 sequence, probably the first thing you can try to do, you can try to overlap, right? And there is sufficient overlap, we can merge it, and we can reconstruct the full CDR3 sequence. So we'll do that using some structure. In this case, I think we were using the suffix tree. Okay, so what will be the example? So that's our G read overlapping only the V gene on the left, and then on the right was the read overlapping only the J gene. And we have shown using some empirical results that overlap of 5 amino acids is enough to conclude that overlap. But we also verify the nucleotide. So behind, we know that those sequencing in the nucleotide space. So we verify that the overlap persists in the nucleotide space as well. So the last step of the algorithm will be to do clustering, just to be aware of the errors and thinking about that in certain cases, we can assemble the CDR3 sequence which contain the errors. So what we're gonna do, we're gonna look at the edge distance between the assembled CDR3 sequences, and if it's will cluster too close, we'll report a consensus sequence across the entire cluster. So as we develop the method, the next step, as we usually do, we do simulations, right? Like we do some sort of benchmarking, validation of the method based on the simulation data. So the reason we do that, as many people in the field, is free cost, right? So if you assume the cluster time is free, so that gives you the free way to evaluate it. But what is nice about that, then you can explore a lot of parameters, right? Like for example, we looked at the coverage. We looked at different coverage as well as different read lengths to ask the question, how our method performs comparable to other methods under different sequencing scenarios. So what you can see here, we have looked at the recall and precision and summarize that as the F1 error as the F score. So across the scenarios, I think IMRAP, our method was performing quite well. But this is the first step, right? So the concern with simulation data we always have, and I think I would encourage everybody to challenge that it's always oversimplification of reality, right? So because here what we assume that doing simulations on the computer will be as good as the sequencing machine, right? But perhaps we will oversimplify reality. So what is also nice to do, if possible, find the gold standard generated through some sort of molecular techniques or through the techniques which are superior in accuracy for the sequence for the approach you're using. So in this case, right, so remember, we have used RNA sequencing, right? And this is what IMREP was performed on. However, if we take what is called the proper BCR-Seq protocol, right, so that will perhaps be technology which can be treated as the gold standard. And this is what we did. So we have found 18 biopsies sequenced by both technology, one which can be treated as the gold standard, and another one which we want to examine. So the first thing we did, we look at the relative abundances of those CDR3 sequences we assembled. And what you can see here on the x-axis, this is us, IMREP, trying to estimate the clonotype frequency. On the y-axis, this is the proper technology estimating the clonotype frequency. So I would say for the majority of the cases, we were quite close in terms of getting the relative abundance. Another step we did was to look at the total fraction as that of the clonotypes or CDR3 sequences we are able to capture. So the way we looked at that, so we looked at the clonotype frequency versus the portion of the repertoire. So you can read it like that. So if all the clonotype frequencies would be larger than 0.06, we'll capture all of them. So you can look at the blue line. This is us, IMREP. So around here, right? Like we're getting all of them, 100%. So, but as the frequency of the clonotype become rare and rare, right? So in other words, there are fewer reads supporting that clonotype, as harder it becomes. And obviously, if there is no reads supporting a particular clonotype, we can do nothing. But this is usually the case for RNA-Seq, right? So because RNA-Seq is the technology which is not properly designed, so that will usually happen that you have very, very few reads. So on average, we are able to capture around 50% of the repertoire in this case of IgH, immunoglobulin heavy chain. So as we kind of developed the method and did the benchmarking, we said perhaps we need to think more carefully about the benchmarking because tissue type or other properties can impact the performance of the method, right? So far, we saw nice results, and perhaps we can say RNA-Seq is great, right? Like can be applied to study the immune repertoire, but is it really the case? And we thought about the cancer research and immunotherapy, that RNA-Seq can be useful there to predict the results of the immunotherapy and predict the progression of the cancer. But first, we need to know is that technology is good enough under different scenarios. So one very important scenario would be to look at the type of the tissue, right? So if the tissue is rich in the T cells, that will mean there are a lot of reads derived from the T cells, because as we sequence, we will sequence proportionally to the fraction of the cells. But if it's poor, will be fewer reads and will be just harder to assemble those clonotypes. So that's one parameter, perhaps. Another one is the type of the repertoire. Are all the clonotypes with the same relative abundance, or are the clonotypes with their different relative abundance? So all of that will impact our results. And we want to make a kind of recommendation here. We want to say, use RNA-Seq in the clinical setting and the cancer research if certain conditions are satisfied. So we want to find those conditions. So, but before that, this is the process called benchmarking. So for those of you not that familiar, so that's a way to estimate, to assess the accuracy of existing tools for a given application. Usually it's done separately from the method development. However, sometimes it's done together, and then it could be problematic because you are the one developing the method and you are the one evaluating the method, so you can end up being in the self-assessment trap. So usually it's recommended the proper benchmarking should be done independently and paying attention to the gold standard data sets being generated, or perhaps the best if you can generate a novel gold standard data sets. And that's also when you develop the method and evaluate it, it's a bit of the stretch, right? So it's like developing the method is already a big task by itself. So bringing benchmarking into play could be too much. So usually it's recommended to do a separate evaluation. So we wanted to coming back to what we are trying to do with IMRAP and RNA-Seq. So we want to ask the question if the tissue type and adaptive immune repertoire type has the impact on the performance of the methods, right? And perhaps can we replace AIRR-Seq technology in the clinical settings? Or in other words, can it be complementary? Because oftentimes when we have big cohorts like TCGA, which is extreme large example, but also like in individual hospital, oftentimes cancer patients are sequenced with RNA-Seq, right? Which gives you the ability to study the gene expression, but perhaps does not give you the ability to study the adaptive immune repertoire. So we want to ask the question, in what cases we can take those RNA sequence and kind of repurpose it, right? And then be confident about the results about the adaptive immune repertoire. So this is the gold standard data sets we have able to have access to. And some of the data we did in the house, in collaboration with UCLA, with the cancer research group. So we got 14 samples in the house, but as well, we got a couple of more samples which were in the public domain. And this is the key to the benchmarking. You want to get to generate, if possible, or somehow get access to the large number of samples across a variety of conditions. So the results of your benchmarking are as robust as you can get them. So if you look at this, so we have a variety of tissue type, including PBMC, skin biopsies, lymph node, intestine, and so on. But also they're different in terms of fraction of the T cells in the tissue, right? So we have data sets where with the high fraction of the T cells in the tissue and the low fraction. So that's nice. As well as we have different Shannon diversity index of the samples, right? So we have some of them being high diversity, some of them being low diversity. So we have quite a bit of combination there, right? Which will perhaps allow us to make those conclusions. So what we mean by, just to be on the same page, so what we mean by low diversity and high diversity, right? So the low diversity will be when you have a particular clonotype which dominates the repertoire, right? So somehow it was proliferated to the degree that it takes substantial space there. So as you talk about high diversity or high SDI repertoire, it's more or less every clonotype has a similar or equal, like more flat profile, right? Like similar frequencies. And if you think for a second about that, it's will be very, very different for the analysis, because if you have the low SDI repertoire, it's kind of enough to capture just one clonotype and know that it's very high frequency, and that will be enough to get an idea about the diversity, right? Like to estimate that SDI. You don't need to know as many low frequency clonotypes. But to get an accurate estimation of Shannon diversity index in this type of repertoire is actually needed to know quite many of the clonotypes. But remember, many of them are rare, they are low frequency, so as a result, it will be hard to do that. So first step we did, we want to be sure we know the fraction of the T cells in the tissue, right? So you would have some expectation. You will say probably the PBMC or the blood will have a lot of T cells there, but we wanted to have a number. So what we did computational deconvolution. So since we have RNA-Seq data, we know the gene expression. And there is a way to go from gene expression into the cell type fraction using the process called computational deconvolution. So this is what we did. And we did a benchmarking of all the methods for the deconvolution before, so we have used our results. So we have evaluated the ability of the methods to compute the cell type proportions from gene expression data on more than 3,000 samples, and as well as on the silica data and in different mixtures of the cell types. So what we saw, we look at the Pearson correlation between the gold standard and ability of the tools, and the tools which we previously developed called GetIt was able to get quite nice across different cohorts, right? So we really wanted to be sure the method is able to handle different type of the data. So in terms of classification, so this is the workflow. So we took RNA-Seq data, we computed the gene expression data using the Selman, and then we went to get it and computed the cell type proportions. And to estimate the T cell fractions, so we took all of them, CD3, CD4, CD8, and kind of summed them up. And if we say it's more than 5%, that will be T cell rich tissue. If it's less than 5%, that will be T cell poor tissue. Right. So this is our samples. So this is the samples according to low SDI and high SDI. So you can see majority of them are high SDI, and for the T cell fraction, very many of them are having the small fraction of the T cell, and some of them have a higher fraction of the T cells. So this is our data, right? So this is the data we'll be working with. So summarizes, so we have kind of 4 types of the repertoire. So we have T cell rich, monoclonal, which is low SDI samples. We have T cell polyclonal, which is high SDI samples, and so on, right? So we have 4 types, and those the tissue type we have for those combinations. So now what we need to do, we need to do benchmarking, where we will, for every type of the repertoire, we'll take all available methods which are able to characterize and assemble the CDR3 sequence there, and we want to know the results for all 4 types. And we want to give recommendations, right? We want to say, if you have type number 1, RNA-Seq can or cannot replace AIRR-Seq, right? So that's kind of the goal. So this is the workflow. So we have RNA-Seq data, which we have processed, and we have either the BAM file available, and many tools, and this is IMRAP, us, but other tools prefer the BAM file, because we would like to get only the unmapped reads, right? So the BAM file gives you ability efficiently to look at the unmapped reads. But some tools prefer the raw data. They prefer to take the raw RNA-Seq data. So this is the mixer. And as we do benchmarking, we have looked at a variety of metrics which are used in the adaptive immune repertoire, and compare the metric which is derived or based on RNA-Seq data and the gold standard. We also play with the sequencing parameters. We try to reduce the number of reads. We try to make the read length shorter. All of that was done computationally, just because we did not want to generate additional samples. But we were really curious to know how the sequencing parameters will affect the results. So those are some results. So this is the low SDI. So this is the monoclonal. And we have T cell rich low SDI and T cell poor low SDI. So the way to read that, so I think the best results, as you can imagine, is the blue one. So it's rich, meaning there are a lot of T cells, but low SDI, also easy. It's that monoclonal repertoire. So what you can see that we can capture all the clonotypes above 0.0003, right? So we capture %. So we capture all of them, right? But as the percentage goes down, as we have more rare clonotypes, we do not capture some of them. But overall, we capture still around 95% of the repertoire. And you can notice here, it was quite interesting, all the methods are very, very similar to each other, right? Despite they do very, very different things in terms of computational, some of them using the graph, or as you saw us, it was quite simple method, just scanning the sequencing. But regardless of different computational approach, they were quite similar to each other in terms of the performance. So another type of the repertoire will be T cell poor, right? There is not many T cells in the tissue, but it's still low SDI, it's still monoclonal. So you can see here that we are this is in orange, right? So we're capturing all the clonotypes above 0.0266%, right? But then it's really goes down, and then overall we capture around 80%, less than 80% of the entire repertoire. Still okay, right? Still okay results. So quite different story is for when you, yeah.
I have a question regarding this types.
Mm-hmm.
So since the amount of sequencing very largely, does also
But
sequence more of them, do you think, in the future?
But can you define what you mean by monosequencing?
Not monosequencing, but just the general amount of data that
Mm-hmm.
And is the reason why we're missing some of these chromotypes also due to the fact that just the underlying data does not have a lot of representations of chromotypes?
Mm-hmm. Yeah, so like as we sequence more, there is a more chance to discover more, right? But there is how should I say? We actually don't know how many clonotypes we know.
Exactly.
But the reason is not kind of related to the sequencing. I mean, it is, but also related to the attempts. There is no attempts to catalog all the clonotypes somehow, right? So nobody attempted like we do in human genetics. We have all the variants sitting on the clean var or whatever the database, right? And some of them are correct perhaps and some are not, but we have cataloged all of them in one place. So that yet to happen for the adaptive immune repertoire. So we don't know how many clonotypes we know. And that will be an interesting question because perhaps it should be saturated. So if we sequence everyone or like a substantial number of people, we will saturate the space, right? So given the length of the clonotype, like we should see all possibilities there, right? But is it the case? We actually
It could also be like that we as a human, for example, just have a very small fraction of possible.
Yeah, exactly. Some of them might not be anywhere, or some of them can be dominant across populations, but we don't know that. We also don't know and this is what I was kind of pointing to that ancestry. We don't know the distribution across different kind of populations, right? And how it's different across populations. So in other words, it's very much in the beginning and of exploration, right? So it would be nice to have that effort, and we try it a little bit, but it would be nice to have systematic effort to catalog all of them and kind of answer your question. Yeah, cool, sure. Any other questions? Okay, so for the low SDI, right? So low SDI, we don't, I mean, it's still low SDI, but we have T-cell rich and T-cell poor. Okay, so sorry, this is here. So now it's a high SDI, right? So more or less every clonotype has the same frequency, right? So then it's harder. So you can see that both for the T-cell rich and T-cell poor, we're doing kind of okay, but not great. And now you can see the results are slightly more different across the methods. So if you look at the red, so that will be T-cell poor high SDI. So you can see that, for example, us, IMRAP, right? We capture all of the clonotype above 0.4%, right? And for the green one, which will be the high SDI, we capture 0.0155%, right? So the results are worse here comparable to the high SDI. I'm sorry, comparable to low SDI, right? So low SDI is easier because it's one clonotype dominant and it's enough to capture that well, and you will get idea already. Okay, so for conclusions here, right, which we did, right? So we have again so we found a good gold standard. We took all available methods and we ran across and we saw the results. So now we need to formulate some sort of conclusions, which we did in our benchmarking paper. So what we said that the RNA-Seq capture most of the TCR clonotype in low diversity samples and can actually also accurately estimate repertoire diversity, which I didn't cover, but this is what we saw, and you can read in the paper that the diversity, right? Like many times in the cancer research, you would like to know the diversity of the sample. So in monoclonal, in low diversity samples, that was done quite well. And what we concluded that RNA-Seq is the good alternative for TCR profiling in T-cell rich tissues and low diversity samples when TCR-Seq is not available. And we perhaps should not go to the T-cell poor samples in general, but especially when it's TCR poor high diversity polyclonal sample, right? Things are just not working there. But the beauty of this, if you think for a second, that we can know that from RNA-Seq, right? Just by looking at RNA-Seq, we can self-evaluate. We can say, oh, that is actually T-cell poor. So, and we will just not go there. Or we'll say that's a T-cell rich and we'll go there, right? So there is that's, I think, the nice thing, that we can put that into the pipeline, which will be saying, which will have some sort of decision tree, where under which condition we can rely and trust the results. So coming back to IMRAP, so applying that on the GTEx data. So GTEx data is post-mortem consortium data coming from around 1,000 of individuals across 50 body sites collected post-mortem. And we have applied, and that comes around 10,000 samples. So we took MRAP, applied that on 10,000 RNA-Seq samples, and this is the results we see. So overall, we got 3.6 million distinct CDR-free sequence, and that's coming to a question like how many do we know? So out of that data, we got 3.6 distinct CDR-free sequences, and majority of them, as you would expect, to be individual specific, right? In other words, they're present in a single individual. However, there is some fraction of them present across other individuals, and we have looked separately at IgH, immunoglobulin heavy chain, and both light chains, kappa chain, and the lambda chain. So some of the clonotypes are shared across many people. So why is that? We don't know yet, but there is some ideas that can relate to the genetics or can be related to the exposure of the population your ancestors are from, being exposed to diverse pandemics, and then that can shape your immune repertoire. So in other words, not every recombination is random, and that's something we anticipate, but we don't know if we don't do population level studies, and this is another reason to do population level immunogenomics to answer those kind of questions. So here what we see is that those are the most public clonotypes, right? You can see a particular one in IgH, immunoglobulin heavy chain, being shared across 58 individuals, and those two clonotypes in the light chains were shared among more than 400 individuals. Yeah.
So if I understand correctly, this is only
yes.
So if we would expand this to also do you think there is a underlying idea of trying to learn what clonotypes maybe present organisms and prevent organisms from being hit by certain diseases or certain
no, it's a good question, right? I think it's more like a response, right? So like adaptive immune repertoire will be responding to a certain disease, right? So it's more like indicative. Like if you have certain clonotypes, can be indicative you're fighting a certain disease rather than predisposition. Because
for example, organisms suffer from, like, this is just a very general example, conditions that they can fight very well. For example, or human immune system
right.
Certain, let's say, have a disease, and I know for somehow this dog does not have the cognitive expect information out of it.
No, it's a good idea. Right. Yeah, you can look at the clonotypes exactly, like absence or the presence of the clonotype relative to disease. Does that, it can be used for humans, right? Like, and because we know that in cancer there is certain clonotypes, can be predictive, or like the shape of the repertoire, or certain clonotypes can be predictive of the immunotherapy result. I mean, outcomes and so on, right? So, or like the kind of the response to the disease and so on, right? So yeah, that's a good idea. Right, engineering them and so on, right? Like exactly. Yeah, absolutely. Sure. So yeah, so this is what we have got, and like I think we did the first attempt to give an atlas of all the clonotypes we have assembled and shared that on the GitHub, but I think going forward will be nice to expand that and bring more sources and give more functionality to the atlas. We also got histology. Yeah.
Sorry,
Yeah, please. Sure, please.
I'm curious. So if you look
right.
of this atlas basically of clonotypes, do you think there would be an underlying structure to clonotypes, meaning, for example, regions where people come from? Let's say they smoke, we not smoke.
Yeah. No, absolutely. Yeah. Like the metadata would be nice. Right. We didn't do that, but I agree. Would be nice to look at the enrichment of the clonotypes, right? Or the structure of the clonotype, because we can look at the motifs, like part of the clonotype enrichment in different groups, right? And the groups can be based on population, based on the lifestyle, based on this. Absolutely, yeah. And that will be, and this is what people kind of see if they sequence different populations. I think it was in Africa, and they see that, like in certain population, which we know were exposed to certain disease, certain number of years ago, we see that clonotype present. In the population which were never exposed to that disease, we do not see that, right? So it's there. But again, we need that large data to do that. And that's like a strong argument why we need data-driven approaches here. That's kind of the only way to look for those enrichments. Mm-hmm. Right.
Sorry, another question.
Mm-hmm.
Based on your knowledge, is, for example, getting this atlas used on metadata with privacy concern?
Yeah. I mean, privacy is one concern, another just incompleteness of the metadata, right? And then I will have slide on that. That if we go kind of, so let me hold on this question. I will have a slide and we'll answer that. I will have right slide to answer that question. Okay, so we also got histology images being available and pathology nodes. So what we use that, some sort of validation. So we have looked at Hashimoto disease in thyroid, and we got a pathology node telling us, that's a late state Hashimoto or early state for Hashimoto. And then what you can see that as the Hashimoto disease progresses, we have more and more clonotype in thyroid. We also look at the sex differences, and we see none, except the tissue you would expect to see in the breast, where the average number of the clonotype in the breast in the female was larger than the male, with one exception of gynecomastia, where the disease where the male breast becomes the shape of female. So we were happy to confirm that case. So I think I will maybe skip this part. We will be very briefly saying that, and we'll move to the future work, and we'll take more questions. We also, as we did that, we realized that all the metrics which are simple, like diversity or the clonality or the number of clonotypes, things like that, they are currently done by the methods which sometimes are not accessible by vet lab or biology researchers. So what we did a Python-based or Jupyter-based notebook tool which can facilitate the analysis in a reproducible way, where you can record all the steps of filtering and other things. So could be, that's I guess separate discussion, but could be more effective than R packages, which are used, if not used in R markdown mode, then like the track of all the changes and analysis are lost. Okay, so I will skip those slides and we'll go to the future work. And this is what the slide good to answer that question about, can we look for the metadata and the privacy concerns. So what we attempt to do, as the next project, we would like to take all the public data we can get access to. And as you can imagine, there is quite a bit. If you think about RNA sequencing data, we got more than 80,000 samples being there for the whole genome data, more than 1 million coming from UK Biobank, coming generally from SRA, coming from Singapore Precise initiative, giving us access to 500,000 whole genome samples. So there is quite a bit there. So oftentimes, or sometimes, we don't know, that the metadata is available. And then some basic metadata like disease status, age, biological sex, things like that, is accompanying the raw omics data. In that case, we can take advantage of that. But obviously, there is some private one which is hard to access or it's not allowed to access. And we hope, looking at initiatives like UK Biobank and Singapore one, there is a standardized procedure to collect metadata, and as opposed to the raw public data, where everyone was collecting metadata differently with different approach and so on. So what we want to do, we want to take all the data and have access to as much metadata as possible and reconstruct the immunome, or make the first attempt to reconstruct the immunome and look at the immunome differences across the populations and find the patterns of enrichments. Do we see certain clonotypes enrich in certain population? Are they associated with certain metadata, like disease status, things like that? So the first attempt we did, so we ran IMRAP on almost on 80,000 RNA-Seq samples on Amazon Cloud. And what we see, we have assembled 12 million more different CDR3 sequencing. And then we also have matching HLA types. We have gene expression data being available to us, which gives us quite a rich resource we are currently exploring. So what we see so far, we have certain number of samples having the disease status being available to us. And we already see some interesting pattern just of the V gene usage. Just V gene usage gives us interesting pattern. And we see different pattern in type 2 diabetes there. So other ongoing immunogenomics project, we also look at the completeness of the database. And in the human genomics, we got lucky and we are 99, I guess, 99 something % complete human genome reference, which happened a couple of years ago. But that yet to be the case for IMGT. So IMGT is the database which is behind every AIRR-Seq analysis. So that's a database of the V, D, and J genes. But it's far away from being complete. And we actually don't know exactly how complete is that. And we don't have a strategy to make it more complete. But it's absolutely crucial because I think it's more profound than in human genomics where certain V alleles will be present only in one population, not present in the other one. So far we have started with the mice and we want to look as the mice and look at the completeness database across different mice type and see if we can bring the sequencing to infer and novel alleles like V alleles and make the database more complete. So this is the data we have able to got from public resources. So we have quite a bit of samples coming from different mice strains. And we have looked so far, we try to assess the completeness of the database by comparing the reads of that particular strain, mice strain, to the database. So what you can see quite a different picture across different mice strains, that in certain mice strains, there are more reads, there are reads with more mismatches. So if you assume more or less equal error rate across the databases, we can attribute the difference in mismatches to the completeness of the database, right? So in other words, the database is serving well certain mice strain and not serving well other mice strains. So similar thing, we would like to expand that analysis and do for the human, look at the different population, and we see the indication in the human that the IMGT database, which is currently state of the art, is not just incomplete, but is disproportionately incomplete across population. And then just because the database was primarily based and built on individual of European descent. So if you take the sample coming from Asian individual, it will match worse. In other words, we'll have more mismatches there. As other future work, we would like to focus on methods to make accurate alignment of RNA-Seq reads. So alignment is very well studied in the human genomics. However, there is not that many attempts to make accurate alignment for AIRR-Seq data. And the data is quite interesting because the reads are usually longer than the genes, right? So it's kind of a reverse problem as we have in the human genomics. And we're exploring some interesting algorithms there. We also would like to develop the method to take to take AIRR-Seq data, whatever the sequencing data we can, and try to assemble not the clonotypes, but take the step back and assemble the novel alleles, right? And if we can do that with the great precision, we can use those alleles and complement the database and make the database more complete. Okay, so that's work of many and diverse funding sources. And yeah, we'll be happy to answer any questions. Thank you.
Hello. So in our group, we've worked on various projects regarding also the traditional genome analysis pipeline, where we accept that sequencing has a very high throughput, but the later pipeline stages are a bottleneck, for example, read mapping or also later variant calling. And for sure, there are also connections when studying immunogenomics in that field. But I was wondering if there could be approaches For example, during sequencing, where you said that we filter based on the T cell count in the cell, that we, after a certain point during the sequencing process, stop the sequencing because we know that, or we estimate that the amount of T cell sequence will be low in that region, so we can continue and save a lot of energy and stuff. Yeah.
Right. Yeah. So the only one thing to consider that, so if you talk about RNA-Seq data, right? So the data is not designed to study the T cells.
Yeah.
Right. So perhaps it's not idea to stop because the data still can be used for gene expression analysis and so on. Right. But you are talking about if you talk about the TCR-Seq or BCR-Seq, right, the proper technology.
Yeah.
So I think that's a great idea because if the sample is monoclonal, meaning it is a single clonotype, you can saturate it quite quickly. And if you don't really need to know about those rare clonotypes in details, but rather have idea about your repertoire, perhaps you can stop quite early, right? You will saturate that. But if you have polyclonal, right, perhaps you need to go further away. So and then that's actually that's great idea, and then I think that's needed because it will save you obviously the cost, and you can sequence more samples, right? Like given the fixed cost of the experiment, you can then adjust the number rather than doing the same number of reads across. You can adjust number of reads depend on the type of the repertoire.
Exactly.
Yeah, absolutely.
Yeah. Sorry, I'm not great with the terminologies. And maybe as a second question, so when moving at population scale, there's for sure, I mean, we have a lot of problems that you have with a lot of data, right? Data movement, so on and so forth. But I do not have an intuitive understanding of what new bottlenecks in this immunogen study pipeline are. It's a very open question. Can you provide some insight maybe that differs from other analysis?
Yeah, no, I think it's like many are the same, right? Like it's the question how we process such a large data, right? As everywhere, if we want to go truly population scale and look at the millions of samples, how we do that, right, in an effective way. So I think so I think in that sense, I don't see anything unique here so far, right? So the same story, we need to have methods which are scalable, which will not require too much computational resources, things like that, right? So it's all kind of the same, right? Because we want really to look at million samples, but the only way to do that to keep the low cost, the cost down, so that we can afford that, right? So because, for example, if you think about that analysis we have run around 80,000 sample, that was not fun in terms of cost. I think it was 50 cents per sample, right? So it adds up. So it adds up really quickly, and I think it's a kind of interesting times now that we really want to look at large data, and we can because data is around. But the question now becomes, can we afford it? And I think the only way to afford it to truly make the algorithm, the methods to be quick and requiring little resources, right? Because that's no longer the theoretical abstract, a theoretical concept, but it's a practical aspect.
Yeah.
Because if your method requires too much memory or runs too slow, you will just not afford to look at the millions, but you will look on the 100,000. But maybe the signal was there, right? So you're kind of restricting yourself.
Can I ask another one? Okay, maybe a last one from me. So you talked about data being disproportionately missing for different populations, which I guess based on the technology provided, so on and so forth, makes a lot of sense. But many other directions and works have a similar problems, right? Especially when you now move to computational medicine, also regarding like machine learning approaches and stuff. So everything is kind of biased. What I was curious is that some of these approaches try, for example, creating artificially data from these undersampled regions to model that basically we have more data from a certain population and so on. Do you think this might be an interesting aspect to think about for capturing more of these? I forgot the terminology, sorry.
CDF-free sequence?
Yeah, exactly. Right. Or if something similar to that would actually hurt you because it would introduce more noise.
Right. No, I think it's like, I would say maybe the very first approach to me would be to mine all public data first, right? Because actually what is happening, the databases are built on Europeans, but the sequencing data is not only Europeans. I mean, sure, it's biased to Europeans, but there's a substantial fraction of different other populations. And mining that data and assemble novel things and complementing the database might be more, as the at least initial step, right? Might be a better approach because that can bring the databases like up to completeness across certain population where the public data exists, right? So that's a very direct, very simple approach, which we should do first.
Okay. Yeah. So the reason it's just because no one has yet shown interest.
Yes, exactly. The reason is just because two reasons. Nobody made an attempt.
Okay.
And two, we actually don't know to what degree that can be accurately done. Can we do it? Because if you think about doing complementing the databases, whatever you derive should be very, very, you can kind of compromise the sensitivity perhaps, but you should really keep precision high because those things will go to the database, right? So in that sense, I think it just was we don't know yet, can we do that, right? Perhaps it's possible, but we don't have numbers yet.
Okay. Yeah. Thank you. Thank you very much.
Sure, absolutely.
We can continue this discussion later on too. Maybe a brief question from me too on the computational side of things. It wasn't entirely clear to me. Does RNA-Seq, the use of it also targets mitigating some performance bottlenecks of the previous methods used?
Can you repeat that?
Like, is it better for cost and computational latency?
Yeah, so the reason we do RNA-Seq because the data exists. So we kind of put out of the equation the cost because RNA-Seq it's currently way more popular data. So we wanted to repurpose that.
Okay.
So that was the whole thing. But if you are deciding, should I go RNA-Seq or AIRR-Seq, perhaps better idea to go AIRR-Seq, right? Because it's a proper technology to do.
Okay, so it's definitely better in terms of accuracy.
Yes, it's definitely better, but oftentimes it's just not available, or oftentimes people prefer to have gene expression data and they go RNA-Seq, and then we say, and then AIRR-Seq will be studying adaptive repertoire will be kind of a secondary thing for that, right?
Okay, so then this brings my second question, which maybe you have already kind of addressed. So then we could use RNA-Seq as some sort of pre-processing fast filtering and then go to the AIRR-Seq if we want more than.
Oh yeah, absolutely. Yeah,
does this exist already?
Right. No, I mean, I think it's like even using the current methods, we can. So if we have let's say we have a set of samples which were done by RNA-Seq, actually can pinpoint us interesting cases which can be further studied by AIRR-Seq.
Exactly.
Right. Okay. So I think the current methods will allow you to do that.
Okay.
Right. So it's actually, So but that's only the case if you need both technologies, right? Like, so you do RNA-Seq on everything, pinpoint interesting cases, and sequence them further with more details using the specialized protocol like AIRR-Seq. Yeah.
That's very interesting.
Absolutely.
Great. And then if no one from the audience has any more questions, there is one online I can read from Zoom. So, can some of the proposed computational ideas also be used in other areas of bioinformatics that face the same challenges, such as data scarcity?
I don't know what for the data scarcity. Yeah, I think so. You mean like repurposing? So yeah, I think in terms of repurposing, I think that can be done for other types of problems like HLA calling and things like that, that we can always be creative. And I think the right word here is creative. So given data available to us, like in this case, we would prefer all the data in the world to be AIRR-Seq and things will be easy for us, right? But that's not the case. So we are creative to repurpose other technologies. So I think other fields will benefit from being creative and try to repurpose the technologies, right? And I think to me here, I mean, sure, it's about computational methods and so on, but also this is about the properties of the data. I think to do that, it's beneficial to truly deeply understand the property of the data and try to understand, does the data can match the question you're asking? Because data was not designed to answer your question, but sometimes it still can. But you need to deeply understand the technological component of that and do proper benchmarking to know that.
That's very interesting. Okay. Thank you very much. Anything else? Furkan has a question. Okay, maybe that will be the last one and we can continue.
So you towards the very beginning, you showed this IMREP's performance across different read lengths, right? And IMREP performed favorably compared to other techniques, such as one of them was IMSeq, I guess.
Right.
And in one specific plot, 50 BP plot, it's performed much better, like compared to others.
Right.
You had more noticeable difference.
Right.
I was wondering why that's the case. Like the others seemed completely infeasible,
Right.
almost.
Yeah, no, it's a great question, and I'm glad you noticed that. So the reason is because we specifically make that second step of IMURAP, where we consider those partial CDR3 sequences. So even the read is not overlapping the entire CDR3 sequence, we can overlap, and none of the other methods do that. They require the read to fully overlap CDR3 sequence. But if the read is 50, it will not do that.
Okay, thanks.
Yeah, sure. Yep, thank you.